being here, where I spend most of my time at the Arnold Arboretum, and for allowing me to share some of the work that I've done in my lab and my thinking of speciation. I am very sorry that Simon couldn't be here, but thrilled to have this opportunity. So as mentioned, I am a faculty member at Harvard, and I work here at the Arboretum. So if you're interested in any of this work and want to actually see these organisms, they're all upstairs, the plants and the butterflies. So my lab thinks about the process of speciation. And I'm not going to define it. Instead, I'm just going to conceptualize this idea that we can know has occurred, where there is a hand-mixed group of organisms that interbreed and share genetic and phenotypic variation, and that somehow over time it splits such that we then have two independently evolving to some degree groups of organisms that interbreed maybe less and have their own genetic and phenotypic diversity. And that's the process that I'm interested in. One framework to look at this in is through the accumulation of barriers to reproduction, which we tend to call reproductive isolating barriers. And we know that there's a lot of forces that influence the evolution of these barriers: natural selection, sexual selection, genomic conflict, drift, geography, obviously. So the work in my lab is largely focused on selection and the role of selection in the accumulation of these barriers to reproduction that we think are then involved in divergence of lineages. So now, in this current scientific climate, and also probably in Darwin's scientific climate, can be argued that the most common way we think about natural selection and speciation is through this process of local adaptation, where organisms evolve, adapt in their environment, and if they're evolving and adapting to slightly different environments, then this can cause barriers to gene flow. And so we get reproductive isolation as a byproduct to standard local adaptation. So we have some really classic examples that have been worked out beautifully, both phenotypically, genetically, measures of selection in the field. We can adapt in the sticklebacks to different salinity environments. We can adapt, aphids can adapt to eating different things. Flowers can adapt to different pollinator environments. Birds can also adapt to eating different things. That's not what I'm going to talk about. So what I'm going to talk about is the process of reinforcement. That is how natural selection can directly favor a trait because it stops hybridization or is involved in this process of speciation. I'm going to define it as an increase in reproductive isolation due to selection to decrease hybridization or costly mating. And to get a better idea of what I mean by this, we'll take a historical perspective. This used to be called the Wallace effect, and sometimes still is. And so a number of people have interpreted Wallace's writings as arguing that natural selection can directly favor the accumulation of reproductive isolation in order to stop diverging lineages from intermixing. So for example, he writes, "It will occur to many persons that, as the infertility," whoops, it's chopped off, "the infertility or sterility of incipient species would be useful to them when occupying the same or adjacent areas. By neutralizing the effects of intercrossing, this infertility might have been increased by the action of natural selection." So in many ways he was not correct, but in some ways he was. And that general concept that natural selection might actually favor lineages not hybridizing and not interbreeding is what he is known for, and what we now know is true and can happen. So this idea was then championed by Jansky during the modern synthesis, and he clarified it and put it in a population genetic context that makes a little more sense to us now. And he argued, where hybridization jeopardizes the integrity of two or more adaptive complexes, genetic factors which would increase the frequency or prevent the interbreeding would thereby acquire a positive selective value, even though these factors by themselves might be neutral. So what he's saying is that if two lineages are evolving on their own separately and then hybridize, if that hybridization is bad, their offspring are sterile or infertile or maladapted to their environment, a trait that actually stops that hybridization would be favored by natural selection simply because it stops the production of those hybrids, and not for any other reason. And this is what we call reinforcement. So the term reinforcement is attributed to Frank Blair, who studied the process in Texas, which is where I study it. I think it happens in other places, but this is pretty neat. So he studied these narrow-mouthed frogs. And he noticed that there were two species that grew in central and eastern Texas. And where they grew apart in Alopatry, they had similar mating calls. And where they grew together, frogs don't grow, where they lived together in Sympatry, they had very diverged mating calls. And he argued this divergence of mating calls reinforced the species boundaries. And it was this divergence that then enabled them to stay separated and not make these maladapted hybrids. So from Frank Blair, we get this term reinforcement, and we see this pattern that has now been classically attributed to reinforcement and of character displacement, where in allopatric populations, species are similar, and in sympatry, they show divergence that decreases hybridization. So divergence in traits related to mating that stops hybridization. So since the modern synthesis, over the next 50, 60 years, we've done an incredible job accumulating examples of organisms that appear to have undergone reinforcement. So we have tens and tens of examples across the tree of life in lots of frogs and toads, many insects, in fish, and even in mammals, and of course plants, where we see traits in sympatry that appear to have evolved to stop hybridization between closely related species. So from this work, we get a pretty clear idea that reinforcement can happen, and that it does happen, and that it might actually be really important when thinking about this process of speciation. But I would argue now is the time to ask maybe more interesting questions. How, why, so what? More specifically, what are the genetic basis of these traits that show reinforcement or that stop hybridization in sympatry? How much reproductive isolation does each gene or trait confer? And what are the mechanisms of selection that allow this trait to stop hybridization and thus be spread in these sympatric zones? So these are the questions that I'm going to tell you about in my research, working on this beautiful Texas wildflower, Phlox Drumondii. So Phlox Drumondii is an annual species. It grows along the roadsides in Texas, and it's part of that beautiful spring bloom that is happening right now. It is pollinated by butterflies, mostly this pipevine swallowtail, Batis philanor. It comes in two color varieties. This is the color of phlox. Almost every phlox species across North America is this sort of light blue purple color. But Drumondii also has this dark red color variety. And what's interesting, or has been interesting about this system, is the geographic distribution of this flower color variation. So most of the phlox range, all the individuals have this light blue purple flower color. But on the eastern populations, all the individuals are dark red. And it's this area of dark red where we see co-occurrence with a closely related species, Phlox cuspidata. So this second species also has that light blue purple flower color that is the same as Drumondii. So it was observed, this classic pattern where you have two species that look alike or similar in allopatric populations, but divergence in sympatry in a trait that is thought to be connected to reproduction and possibly hybridization. So Don Levin in the 80s observed this pattern and hypothesized that indeed this change in flower color was a result of reinforcement or selection pressures to stop hybridization between these two species. And we have a bit of evidence before I started working on this system that this was likely the case. So we know that hybrids do form in nature. This is Phlox stromundii, the dark red form. This is Phlox cuspidata, this light blue, with the light blue flower color. And this is a punitive hybrid shown in a mixed population. So they grow together and they do produce hybrids. Those hybrids have low fertility. So sterility in one direction of the cross is about 100%. Sterility in the other direction is 25 to 50%. So there's a cost to making these hybrids. They're very pretty and they grow great, but they're not so good at making offspring. There's also some preliminary evidence that these dark red flowers actually don't hybridize as much as the light blue flowers. So the research that I'm going to tell you about is investigating first the genetic basis of this trait. And the trait we're interested in is flower color variation within a species. And how much reproductive isolation is conferred by this flower color change. And then what are the mechanisms of selection that allow flower color change to actually decrease hybridization between the species. So in order to look at the genetics of this trait, we can do classic genetic crosses. Again, it's variation within a species, so we can cross the light blue variety with the dark red variety. The offspring, F1 offspring, are this beautiful deep purple, which I call dark blue flower color, and self those individuals, and we get 4 distinct flower colors. We regain this light blue and the dark red. We get the F1 color, this dark blue, and we get a new color, light red. And these 4 flower colors appear in a beautiful 9 to 3 to 3 to 1 ratio, showing us, 1, that Mendel is right, and 2, that it's a very simple genetic basis, where there's just 2 loci. One locus controls flower color hue, and this controls the type of anthocyanin pigments made in the flowers. So like most flowering plants, these make anthocyanin pigments in their petals, and that gives them the color. These blue individuals here, which I'm calling blue, I know they're purple, but I'm calling them blue because they make both red and blue pigments. These red individuals here only make red pigments. So we call these blue, these red, and that's what differentiates individuals at the hue locus. The second locus corresponds to the amount of pigment made in the flower. So these produce a little bit, they're light. These produce 3 times as much, they're dark. So in allopatry, we have light blue individuals. In sympatry, we have dark red individuals. And you get there by going from light to dark, from blue to red. So what are they? Well, I told you they're anthocyanin pigments. And those of you who know plants, well, maybe if you don't know plants, you know that the anthocyanin pigment biosynthesis pathway is one of the most beautifully conserved genetic pathways around. And so we can use, we can sequence these genes that are part of this anthocyanin pathway from just about any plant you want. So I could use a candidate gene approach. This is the pathway, and sequence all of the genes involved to look for what gene actually causes flower color change. So if we look at this pathway, in bold, you're going to see the enzymes that are actually involved in the production of these pigments that are at the bottom, the red ones and the blue ones. In this box are transcription factors that in other species have been shown to regulate these genes. So all I did was sequence all of these genes from light blue and dark red parents of my cross, found markers, SNPs that differentiated them, the 2 parents, then genotyped my F2 offspring, all of those flower colors, and found an association at the genetic level between a marker in a gene and the flower color in the plant. And what did I find? I found perfect associations between genotype and flower color phenotype for 2 genes. So when we think about the hue locus, again, this hue locus controls either you produce blue pigments or you don't. And that is perfectly associated with the genotype of F3'5'H, which is the branching enzyme that turns the precursors of the red pigment into the precursors of the blue pigment. So it turns out, if you don't express F3'5'H, you don't make blue pigments. And that's exactly what's going on in these flowers. These don't express F3'5'H, and these do. The second association, genetic association, was with the transcription factor, this R2R3 MIB. And so what we showed was that in these dark individuals, the R2R3 MIB transcription factor is upregulated, thus upregulating all of these enzymes in this pathway, and you make a whole lot more pigments. So that was all a very complicated way of saying this flower color change is caused by 2 genes, one that affects the amount of pigment that's produced, I'm calling that the intensity locus, and the second affects the type of pigments produced. That I'm calling the hue locus. So now that we know the genes involved, and I would argue more importantly, we know there are only 2, we can ask if either or both of these actually affect reproductive isolation in this system. And if so, how? So next I'm going to tell you about whether light blue or dark red or any of those other colors that can be produced actually hybridize less or more with Phlox cuspidata. So it was hypothesized that in this region of sympatry, we had the evolution of dark red flower color in order to stop this hybridization. And I would argue that the reinforcement hypothesis, this idea that a trait can evolve simply because it decreases hybridization, has 2 parts. One, without the second species, all the flower colors, all the genetic alleles are equally fit. So you might imagine that fruit set for light blue, light red, dark blue, and dark red flowers are all the same, grown together in a common garden without cuspidata, the second species. But with the second species, the reinforcement hypothesis predicts that a decrease in hybridization selectively favors the dark red flower color. So for example, if this grayed out part represent the hybrid fruits, and since they're pretty much sterile, they don't count towards fitness, then you would see that the dark red have fewer hybrids and therefore are at a selected advantage. So this is what we want to test in this system. And we want to know, is it because you're blue or red, or is it because you're dark or light? So I did just that field experiment, where first I put all 4 of my flower colors together in a common garden environment without Phlox cuspidata, planting 3,000 individuals, letting them grow, flower, set fruit naturally, counting all of those fruits, looking at survival, and finding that they all did equally well. So there was a lot of variation, but none of it corresponded to flower color genotype, thus indicating that these 4 flower colors grown in that sympatric environment do equally well in the absence of the second species. So what about if we put cuspidata there? Do either of those loci actually decrease hybridization? Again, we did this as a field experiment where I put each of those 4 flower colors of Drummondii in a common garden with Phlox cuspidata, the second species. And again, they grew naturally, they flowered naturally, the pollinators came, pollinated naturally. I collected the fruits from the Drummondii plants, grew them in the greenhouse, genotyped those offspring at a genetic marker that differentiated cuspidata from Drummondii. And with this paternity test, I could then estimate hybridization rate for light blue, dark blue, light red, and dark red flower colors. So hybridization was allowed to happen naturally in the field, in this controlled sympatric environment. What did I find? First, I found there was a lot of hybridization. So when I grew, this is the allopatric Drummondii color that looks like the cuspidata color, and 38% of the offspring were hybrids. So that meant almost 40% of those offspring produced by this plant are sterile. This indicated to me that hybridization could indeed be a huge selective force controlling the evolution of traits in this system. What I want to know is if the dark allele or the red allele actually decrease this 40% hybridization rate. And it turns out it does. So plants that are dark or have even just one copy of that dark allele have a 50% lower hybridization rate. So dark blue and dark red plants hybridize at about 15 to 20%. Light blue and light red plants hybridize at 35 to 40%. So now by understanding these genetics, we know that there is one locus, one gene, that decreases hybridization by 50% in this system. And we've measured reinforcement actually occurring in a natural environment. If we look at selection on this, now this is in a controlled environment where I controlled the ratio of the plants that are grown. But indeed, based on the fruit set of these plants and the proportion of hybridization, that is the sterile seed production from hybrid pollen, we can see that there's very strong selection favoring the dark allele in this sympatric environment. So it looks like in this system, indeed, at least one of those genetic changes does significantly decrease hybridization. And that this decrease in hybridization is the only measurable selection on that locus, mechanism of selection on that locus. So how does it do it? Maybe not surprising, in order to figure out the mechanism of selection in this system, we have to look to the pollinators. So I told you that the main pollinator is this Batis philonore, this pipevine swallowtail. It accounts for 80 to 95% of the visits to these plants. Turns out it visits all of the plants. It visits the light blue, the dark red Drummondii, and it visits the cuspidata. There are some skippers in Texas that also visit these plants, and once again, they visit all of them. So it's not that classic story we often hear about in plants where you go from blue to red and you switch pollinators from bees to hummingbirds. Instead, that would have been nice. But instead, we keep the same pollinators, but still get a reduction in hybridization. So how does that happen? For this, we have to turn more to animal behavior and think about the movement of these pollinators between individual plants. So to think about this and to experimentally look at this, I did a big potted plant pollinator observation experiment where I took plants of Drummondii and cuspidata and I alternated them in a big array. And within the Drummondii, I alternated the light blue and the dark red, or the dark blue and the light blue. And I watched how an individual pollinator came to the array and moved between different individuals, and thus moved pollen between different plants. So there's 3 array types, one of them looking at the effect of intensity on pollinator movement, and this includes the light blue and the dark blue plants. One of them looking at the effect of hue on pollinator movement, so including the light blue and the light red Drummondii with cuspidata. And one of them looking at both, so the light blue and the dark red, both alleles are varying in this case, with cuspidata, seeing how pollinators move. I'm going to show you my results in this format. So what you're looking at here are the percent pollinators that went from a light blue individual and then next went to a cuspidata, so moved between species for light blue and light red. So that's the hue array. And as you can see that percent transitions between species is the same regardless of flower color hue. So it appeared that nothing about being red or blue changed the way pollinators move between species. And it's the same in the other direction. So a pollinator that was on a cuspidata plant, visiting a cuspidata plant, and then went to light blue and light red at equal transition rates. So what about the dark? It's a very different story. So it turns out when pollinators are on a dark blue Drummondii, they don't tend to go to a cuspidata plant next. So the percent transition between species significantly decreases when the Drummondii flower color is dark. And that's true if they're on a cuspidata. Again, they're much more likely to go to a light blue Drummondii than a dark blue Drummondii. And the same is true when Drummondii is dark red. So this is the sympatric color. This is the color we see all over East Texas right now. Turns out pollinators don't tend to go between the dark red Drummondii and the cuspidata nearly as much as they go between the light blue Drummondii and the cuspidata. So what is this? This is a pattern that pollination people call constancy or fidelity, where a species goes to everything, but an individual in a particular foraging bout will go from dark red to dark red to dark red to dark red, and a different individual of the same species will go from light blue to light blue to light blue to light blue and not care or not differentiate between the two species. And so when it goes from light blue to light blue to light blue, it moves pollen between Drummondii and cuspidata, and you get hybrids. So importantly, these hybridization results from the field experiment align perfectly with the behavior results we observed in these potted plant arrays, where indeed we have rampant hybridization when Drummondii has these light blue flower colors, and we see lots of pollinators moving between Drummondii and cuspidata. But when Drummondii is dark, we have less hybridization, and we can observe this pattern of constancy where pollinators don't tend to move between species. So we should be happy with that, right? I was pretty happy with that. We've got a mechanism of selection in that sympatric zone that seems to make sense and explain why we're dark red here. Okay, I have one more problem for you. Why are we light blue here? If there's such strong selection on dark red, and I'll tell you now, there's lots of migration within Drummondii across this whole range, why have we not swamped out this light blue? So this is something that's come up a lot in reinforcement, is why do you still see this pattern of character displacement? Why do you not get a uniform dark red Drummondii if there's such strong selection for it? So I wanted to look at that as well, and since I was looking at butterflies, we thought maybe the butterflies would be important. So in order to look at this question, we created arrays with just Drummondii plants, but we alternated two different colors, the light blue, dark blue. Maybe pollinators also have a preference for light over dark. We also had arrays that looked for preference based on hue, where we have light blue and light red plants. And finally, we looked to see if pollinators showed preference for light blue versus dark red. And this was an effort to understand what is actually maintaining that light blue flower color in these areas of allopatry. So these are how I'm going to show you the results, where we have proportion of visits with the 50-50 line right here. Anything above it is preference for light blue. So indeed, in our arrays, we found that pollinators way over visited the light blue plants relative to the light red plants. This strong preference for blue over red was highly significant. Turns out the pollinators, these butterflies, also prefer light over dark. And when you put dark and red together, the pollinators don't like it at all. They visit twice as many light blue flowers as dark red flowers. So I think looking at these pollinators has really told us why we get light blue here and dark red there, and can start to explain the selective environment on flower color across the entire range. So my work in this system has shown that for one case, we have a simple genetic basis to a trait that is involved in reinforcement. And that at least one of those loci involved significantly decreases hybridization. What the other locus does, no idea. It's a good thing I have a career ahead of me. If you have any ideas, let me know. We also have found out that you have to look beyond your system and actually branch out maybe to animal behavior. As a botanist, that's sort of fun. To think about what is the mechanism of selection and how this works. And for this, we actually turned to the pollinators and found that their constancy behavior explains the patterns of hybridization in this system. So I hope this little case study is an example of where our research is going now. I would argue that we started asking, the general scientific community, Wallace, started asking, could natural selection favor speciation? Then we went on to asking, does natural selection favor speciation in this direct way? And for 50 years, we found many, many, many examples. And now I think we're at a beautiful point to start asking how, why, and so what? And begin to incorporate genetic and genomic tools, advanced statistical analyses, more resources to do field experiments to really get at how how natural selection actually influences the evolution of lineages through directly favoring speciation. And with that, I will thank collaborators, funding sources, and of course, field sites for their assistance. Thank you.
Thank you for a wonderfully clear